Okay, so welcome to this next video in which we're discussing the drug treatment of rheumatoid arthritis. Okay, so we're currently about to look at the mechanisms of azathioprine and mycophenolate, which are both very powerful immunosuppressive agents which achieve their action by preventing proliferation within B and T lymphocytes. Okay, so before we discuss the way that they work, we need to discuss the structure of DNA, and I think when discussing the structure of DNA, it's very useful to discuss all of the terminology. So I'm just going over some of the terminology that we're going to use to understand the mechanisms of azathioprine and mycophenolate. Okay, so we've discussed that a nucleoside is this name for a ribose sugar here with an organic base bound off it. So here is the ribose sugar in blue here, and then we have an organic base coming off the side here in green. And the organic base can be one of these 5 here, although there are more, okay, but these are the most famous organic bases. Okay, then we need to distinguish this between a deoxynucleoside. So a nucleoside has ribose. A deoxynucleoside is deoxyribose with an organic base, and the difference between deoxyribose and ribose is that deoxyribose does not have an alcohol group coming off the second carbon here. Okay, so remember, the sugar phosphate backbone of RNA molecules contains the sugar ribose, and the sugar phosphate backbone of DNA molecules contains the sugar deoxyribose. Okay, and that's going to become important later. Right, we then want to discuss what a nucleotide is, and a nucleotide is kind of this loose term basically, because it could refer to a structure that we've shown here, which is an organic base here in green, okay, with a ribose sugar coming off it. And it's important to understand that, again, nucleotides refer to ribose, okay, not deoxyribose. Deoxynucleotides then refer to deoxyribose. And then finally you have phosphate groups coming off, but what's woolly about this is that it doesn't tell you how many phosphate groups coming off. So it could have a single phosphate group coming off, and the strict name for that would be a nucleoside monophosphate. Okay, so a nucleoside with a single phosphate, or it could have 2 phosphate groups coming off, in which case it would be a nucleoside diphosphate. Okay, and again, nucleoside diphosphates would be considered a nucleotide. Okay, and then finally it could have 3 phosphate groups coming off, in which case it would be called a nucleoside triphosphate. Okay, so basically that's why the term nucleotide is a little bit difficult, because it could mean a nucleoside monophosphate, it could mean a nucleoside diphosphate, or it could mean a nucleoside triphosphate. And by the way, nucleoside monophosphates are often abbreviated to NMPs for short, nucleoside monophosphates. Nucleoside diphosphates are often called NDPs, and then finally nucleoside triphosphates are often abbreviated to NTPs. Okay, right. Finally, there is then also deoxynucleotides. Okay, and deoxynucleotides would refer to a deoxynucleoside. Okay, so where you're incorporating deoxyribose rather than ribose into the nucleoside, and then with a certain number of phosphate groups on. So again, there would be deoxynucleoside monophosphates, deoxynucleoside diphosphates, and deoxynucleoside triphosphates, and these would be abbreviated as DNMPs. That would be deoxynucleoside monophosphates. Then DNDPs, which would be deoxynucleoside diphosphates, and then DNTPs, which would be deoxynucleotide triphosphates. Okay, right. One last little thing we need to discuss is the names for the nucleosides. Okay, so basically, remember we've discussed that a nucleoside is ribose plus an organic base. Well, basically, for each of the organic bases, the nucleoside has a name. So when you take ribose and stick on the guanine organic base, the name for that nucleoside is guanosine. Okay, so that's what guanosine means. It means guanine plus ribose. Now if you wanted guanine plus deoxyribose, you would call that deoxyguanosine. Okay, then cytosine. If you take cytosine and bind it to ribose, the nucleoside that you get is known as cytidine. Okay, then if you take thymine and add it onto deoxyribose, the terminology is slightly more difficult here. Okay, so it's not called well, its name doesn't look anything like thymine, basically. Instead, it's called 5-methyluridine. Okay, and you'll see the logic for that in a moment. Okay, so uracil and thymine are very closely related. In fact, another name for thymine is 5-methyl-uracil. Okay, so that's why when you take thymine and add it onto ribose, you call that 5-methyluridine rather than anything to do with the name thymine. And the reason that you don't use thymine here is because we are talking about the nucleoside. We are talking about thymine bound to ribose. Okay, now thymine is not used in RNA. Okay, so it would be rare to see thymine bound to a well, you do see it obviously in cells because it's a precursor to the deoxyribose form, but basically this name where you use thymine is reserved for the deoxynucleoside, basically, and I'll show you this in a moment. Everything will become clear. So guanosine means guanine plus ribose. Cytidine means cytosine plus ribose. 5-methyluridine means thymine, also called 5-methyl-uracil plus ribose. Adenine plus ribose, the nucleoside, is then called adenosine. Okay, and finally, uracil plus ribose is then called uridine. Okay, so these are the names for the nucleosides. Okay, ooh dear, that's smudged nasty. I can sort of cover it up though. Okay, so these are our nucleosides. Okay, now, when you convert them into the deoxynucleosides, most of their names convert very simply. So guanosine, if you wanted to create the deoxynucleoside, i.e. you wanted to create guanine bound to deoxyribose, you would just call that deoxyguanosine. If you wanted to create the cytidine deoxynucleoside, then what you would do is you'd call that deoxycytidine. If you wanted to create the deoxynucleoside of 5-methyluridine, you do not call that deoxy-5-methyluridine. Well, you could. People might know what you mean, but there's another name for this. Okay? It's then called thymidine. So thymidine does not mean thymine plus ribose. It means thymine plus deoxyribose, and that's an important thing to understand. If you mean thymine plus ribose, you must call it 5-methyluridine. Okay, and the reason, remember, is that thymine is only found in DNA. Okay, so it would be more normal to think of it bound to deoxyribose rather than ribose, so that's the reason for the naming difference there. Okay, and then adenosine, if you wanted to create the deoxynucleoside of adenosine, it's called deoxyadenosine, and again, if you wanted to create the deoxynucleoside of uridine, it would be deoxyuridine. Okay, so the naming is all simple from that. And then if you want to then create the nucleotide, you would call it, for instance, if you wanted guanine bound to deoxyribose bound to 3-phosphates, that would be called deoxyguanosine, which would be the guanine bound to the deoxyribose, and then you would just put triphosphate after it to show you how many phosphate groups you have. So we now know how to handle the organic bases, how to handle the sugar that it's bound to, whether it's bound to deoxyribose or ribose, and then we know how to handle putting the number of phosphate groups that you've got bound on. Okay, right, so we now want to talk about the structure of DNA, and then how you replicate DNA, and also how you produce mRNA. Okay, right, so basically, let's start off with the structure of DNA. So, the structure of DNA, basically, DNA consists of deoxynucleoside monophosphate nucleotides. Okay, so here is the deoxyribose sugar here with a phosphate group, and then it will have an organic base, and I might just make this organic base guanine. Okay, and they polymerize together, so you'll have another one underneath here. Okay, so here is the next nucleotide along. And of course you can call them nucleotides, and it's very common to call them nucleotides. Okay, because of course deoxynucleoside monophosphates are a form of nucleotide, though of course they should really be called deoxynucleotides rather than nucleotides. Okay, so we'll then make this organic base thymine maybe. Okay, now this process can continue on, so you can polymerize on and on and on, joining more deoxynucleotides on. Okay, but then the other strand will be here, and it will be complementary, so the organic bases that you have will be complementary to the organic bases here. So you'll have a cytosine here, and then there are 3 bonds between guanine and cytosine. Okay, so I'll put those there. And then here you'll have an adenine, and there will be 2 bonds between adenine and thymine. Okay, and then the sugar phosphate backbone of this other strand runs anti-parallel to the sugar phosphate backbone of this strand. Okay, and what that means is that it's oriented in the exact opposite direction. So as you can see, the pentagons here of the deoxyribose sugars are in the opposite direction. They're pointing in the opposite direction. Okay, and then this will be bound there. Then you'll have your 5th carbon up there with the phosphate group off. So this is the structure of DNA. So you have these polymers of deoxynucleotides bound to one another, and then you'll have 2 complementary strands which will then be joined together to make the double strand of DNA. Okay, right. Now, when you replicate the DNA, what happens is that you pull the 2 strands apart, and of course both of those strands are then, they both carry a copy of the genetic code. Okay, so because the 2 strands are complementary, you can work out what the other one needs to be. So actually, in order to produce a piece of double-stranded DNA, you don't need both strands. You only need one of them. So if we split the 2 apart, like so, we can actually produce perfect copies of the other strand that you've just lost. Okay, so if I call this strand 1 to help me explain this, and let's call this strand 2. Okay, so strand 1 has now lost strand 2, but strand 1 contains all the information it needs to make an exact copy of strand 2, because all it needs to do is make a strand that has exactly complementary organic bases, and then it's done. It's made an exact copy of strand 2. Okay, so I'll call this, I'll colour it in blue there. Okay, and strand 2 again contains all the information it needs to make a copy of strand 1, which it's just lost. Okay, so it again can synthesize a new strand. Okay, so this is how you replicate DNA. Now, in cell division, you are going to need to replicate DNA. Okay, so cell division, remember, is the process by which a cell goes from being 1 cell to being 2 cells. Now, in the process of cell division, you're going to have to copy all of the 46 chromosomes, because this cell has 46 chromosomes, and each of the daughter cells is also going to need a copy of these 46 chromosomes. So you have to copy each one of these 46 chromosomes, and this is how you do it basically. Each chromosome is a massive, great double-stranded piece of DNA. Okay, and each one you're going to replicate like this. So you could view this as being an entire chromosome, and each one you're going to replicate like this, where each of the daughter strands ends up with one of the strands of the original double-stranded piece of DNA, and then also ends up with a new piece of new strand of DNA. Okay, and this is what's known as the semi-conservative model of replication. Okay, so this is semi-conservative replication, and it's called that because you're half conserved, basically. If you look at this daughter strand here, half of it is from the original one, so half has been conserved and half is new, basically, so half wasn't conserved, and that's why it's called semi-conservative replication. Okay, right, so how do you actually go through this process of producing this new strand? Well, if you can imagine pulling these two apart, let's show this on the other page. Okay, so get a new piece of paper. So, what's happened now is we've pulled these two strands apart. Okay, so I'll show this here. And what you're going to do is you're going to produce these new strands, basically. Now, it is quite a complicated process, and we're certainly not going to go through it in a huge amount of detail. Okay, so I'll draw one of these strands that we've pulled apart. Okay, so remember we just drew two deoxynucleotides here, so here are the sugar phosphate backbones, and the two organic bases that we had used on this strand were guanine and thymine. Okay, so let's put that here. So we'll have guanine here, okay, and thymine here. Now the enzyme DNA polymerase actually can only add on a deoxynucleotide when it's already got a strand to add it on, and this is quite complicated. So basically what happens is you have to produce RNA primers, but we're not going to worry about that. So we'll just imagine that we have started the process of replication, and I'll show you how you elongate. Okay, so let's say that we have synthesized. Okay, imagine that the rest of the strand continues on here, and let's say we have already synthesized up to this point here. Okay, so we've already begun the process of replication, and we're not going to discuss all of that. Okay, and we're now looking at how we elongate it. So I'm just going to draw on the one that should be here, and then we'll look at how we add on this final one here. Okay, so the phosphate group should be there then. Okay, and it should then come down like so, and I'm shrinking it all in just so that it can fit in, and that shouldn't be going like that, wrong angle. Okay, it should be coming down like here, and this should be coming up like that. Okay, so here's our deoxyribose sugar. Ignore that line that I've put in there, that's a mistake. Okay, and then coming off here you then have the organic base adenine, and between the organic bases adenine and thymine you then have two hydrogen bonds. Okay, then coming off this, oh well, actually not coming off this yet. Right, so let's say we now want to add on the next nucleotide, the next deoxynucleotide here, basically. Basically, what you bring in is you bring in a nucleotide, a deoxynucleotide that has 3 phosphate groups. Okay, so you're going to bring in the deoxynucleoside triphosphate. So let me show this. So, what's going to come in and sit here? Here is the deoxyribose sugar. Okay, here is the organic base coming off the deoxyribose sugar. Okay, and the organic base that's complementary to guanine is cytosine. Okay, and they'll form 3 hydrogen bonds. And originally this will not just have a single phosphate group coming off this 5th carbon. Instead, it will have 3 phosphate groups coming off. Okay, so you bring in the deoxynucleoside triphosphate, and this is specifically deoxycytidine triphosphate. Okay, cytidine triphosphate. Okay, right. Now you only need one of those phosphate groups to make the sugar phosphate backbone. So what then happens is the enzyme DNA polymerase, and it will be DNA polymerase free, but don't worry about that. What it's going to do is it will cleave this bond between this first phosphate group and this second phosphate group. And by the way, when we're talking about phosphate groups on nucleotides and deoxynucleotides like this, it's customary to call the first phosphate group alpha, the second phosphate group beta, and the third phosphate group gamma. Okay, so it's going to cut the bond between the alpha and the beta phosphate groups, and what will happen is the beta and the gamma phosphate group will remain bound together and come off like this. and will form a molecule known as pyrophosphate. So when you have two phosphate groups linked together, that's called pyrophosphate. Okay, and then what will happen is this alpha phosphate group will link up to this third carbon down here. This third carbon has an alcohol group coming off it, even in deoxyribose, and it will form a phosphoester link with this alpha phosphate group, and basically you've then added on the next deoxynucleotide onto your growing DNA strand. Now the process is pretty much the same for the production of mRNA. Okay, what will happen is, if we draw a cheaper picture of the DNA strand, what will happen is the DNA will have to open again, okay, but this time only one of the strands will be used. Okay, so remember, RNA is a single-stranded molecule. Okay, so let's call this strand 1 and this strand 2. Now, let's say strand 1 is the one that's going to be used. The one that is going to be used as the template for the mRNA is called the coding strand. Right, now what's going to happen is you're going to create a portion of mRNA that is complementary to this portion of DNA. Okay, so this is the new thing that you've created here. And you're not going to use strand 2, which is the non-coding strand. Okay, this piece of mRNA will then come off here and go into the cytoplasm and be translated to make a protein. Right, now the process of producing that mRNA is very similar to how you would elongate a DNA strand. Okay, the difference is that the sugar you will be using will be ribose rather than deoxyribose, and secondly, you won't put in thymine organic bases, you'll put in adenine organic bases. But the process of bringing in the nucleoside, for it will be a nucleoside now because we're dealing with ribose here rather than deoxyribose. The process of bringing in the nucleoside triphosphate and then cutting off the beta and the gamma phosphate for the alpha phosphate to then bind to the third alcohol group of the ribose sugar below it, that will be exactly the same, and you'll produce an mRNA strand there. Okay, so basically, in order to make new copies of DNA and also to make mRNA, you need these nucleoside and deoxynucleoside triphosphates. Okay, so the way that azathioprine and mycophenolate are going to work is they're going to stop the synthesis of certain members of these deoxynucleoside triphosphates and these nucleoside triphosphates, and thereby they're going to stop the production of mRNA and also the replication of the DNA. Now, production of mRNA is also very important for cell division, the reason being that if a cell is to divide from being one cell to being two cells, then it needs to produce a whole host of new proteins, basically, because it's going to go from being one cell to being two cells. So all of the essential enzymes, for instance, all of the enzymes that are involved in the respiratory reactions, and all of the receptors in the membranes, and all of the signaling proteins that act downstream of the receptors, all of these are going to have to be duplicated because you have enough at the moment for one cell, and you need to split those between two cells, and basically there's not going to be enough, so you have to make new proteins. Now, if you cannot produce mRNA because you don't have the correct nucleoside triphosphates to produce the mRNA, then you're not going to be able to translate that mRNA into proteins, and therefore you're not going to actually be able to make proteins. So you're not going to be able to make the new proteins that you need in order to go from being one cell to being two cells. Okay, so for both of these reasons, firstly, because we're going to stop the synthesis of the deoxynucleoside triphosphates, which will stop us from replicating the DNA, and also because we're going to stop the production of the nucleoside triphosphates, which will stop the production of mRNA. both of these are going to then stop cell division, and they're going to do it specifically in T and B lymphocytes. Okay, so how does azathioprine and mycophenolate, how do they work? How do they stop the synthesis of certain nucleoside triphosphates and deoxynucleoside triphosphates? Okay, so we'll start off with azathioprine then. Okay, so azathioprine is going to work by blocking an enzyme known as amidophosphorybosyl transferase. Okay, so this is a big name. So the target for the drug azathioprine, okay, is that it's going to azathioprine is that it's going to block this enzyme, which is called amido, and then it's phosphoribosyl transferase. So amidophosphorybosyl transferase, and this is abbreviated usually to ATAs. So all of the amidophosphorybosyl is abbreviated to a single A, and then the T is for the transfer, and then the A's is for the A's that's on the end of every enzyme. So amidophosphorybosyl transferase is often abbreviated to ATAs for short. Okay, now what is this enzyme going to catalyze? What's its usual function within cells? Well, basically, let me explain basically what it's going to do is it's going to usually catalyze a reaction where it converts a molecule known as alpha phosphoribosyl pyrophosphate, and it's going to convert it into 5-beta phosphoriboxylamine. Okay, now to understand this reaction, I need to explain to you the structure of ribose in a bit more detail. Okay, so strictly speaking, when people talk about ribose, okay, when we talk about ribose in RNA and DNA, we mean the cyclic form of ribose. Now, basically, there is a linear form of ribose and there is also a cyclic form of ribose. Now, strictly speaking, if you say ribose, you mean the linear form of ribose. To talk about the cyclic form of ribose, what you are meant to say is ribofuranose. Okay? However, everyone all the time just calls it ribose. However, strictly speaking, when you talk about the cyclic form of ribose, you really should say ribofuranose. Okay, now there are different isomers of ribofuranose. Let me explain these now. Okay, so let me show the skeletal structure of ribofuranose. So, basically, it is a 5-membered ring, like so, where we have 4 carbon atoms here and a single oxygen atom here. Now, off this carbon here, off this carbon here, and off this carbon here, you then have alcohol groups, and off this carbon here, you then have the 5th carbon coming off. Now, if you imagine that this ring is within the plane, then basically, if we imagine looking at this carbon here, this carbon has one interesting group coming off it, which is the 5th carbon, but it also has a hydrogen coming off it. Now, either the hydrogen can go into the page and the 5th carbon can come out of the page, or the 5th carbon can go into the page and the hydrogen can come out of the page. So we're now talking about optical isomerism, basically. Now, usually, the form of ribose that is in DNA is what is known as D-ribose, or D-ribofuranose, since we're talking about the cyclic form. Now, in D-ribofuranose, the 5th carbon comes out of the page towards us, okay, and the hydrogen goes into the page away from us. There is also what is known as L-ribofuranose, which would be where this 5th carbon goes into the page away from us and the hydrogen comes out of the page towards us. Okay, so here's where the 5th carbon is, and then it also has an alcohol group coming off it. Okay, right. So that's the optical isomerism of this carbon here dealt with. Okay, now, because we're talking about ribose, the optical isomerism of the second and third carbon are dealt with instantly. Okay, so each of these two carbons has an alcohol group coming off it and also a hydrogen coming off it. Now, on both of them, the alcohol group is going to go into the page away from us, and the hydrogen is going to come out of the page towards us. So again, on here, the alcohol group goes into the page away from us, the hydrogen comes out of the page towards us. Okay, if you changed that, i.e. if you brought this one coming out of the page towards us, it would no longer be ribose, basically. It would be some other aldopentose. Okay, right. Finally, the alcohol group coming off here is debatable, basically. This is something that arises as soon as you start making cyclic ribose. So it's never a problem if you're just talking about the linear form of ribose without the ring. However, as soon as you make ribofuranose, suddenly the alcohol group that comes off this first carbon here can either come out of the page towards us or it can go into the page away from us. Okay, and there's also a hydrogen coming off here as well. So if the alcohol group goes into the page away from us, the hydrogen will come out of the page towards us, and if the alcohol comes out of the page towards us, the hydrogen will come out of the page the hydrogen will go into the page away from us. Now, if you just write D-ribofuranose, it does not tell you which direction the alcohol group goes in. So what you would have to do is draw this squiggly line to say it's either going into the page or it's coming out of the page. However, there are two separate optical isomers of D-ribofuranose. If the alcohol group goes into the page away from us and the hydrogen therefore comes out of the page towards us, that would be called alpha D ribofuranose. Okay, whereas if the alcohol group comes out of the page towards us, like so, off this first carbon, that would be called beta D ribofuranose. Okay, and these two optical isomers of D-ribofuranose are known as the anomers of D-ribofuranose. Okay, and the reason that they're called the anomers, okay, is that this first carbon here, and let me highlight it in the colour. Okay, so this first carbon here in green, this is known, oops, let me just straighten this up. Okay, this first carbon here is known as the anomeric carbon. Okay, so these are then called anomers. So you have two anomers of ribose, specifically of D-ribofuranose. You have the alpha D-ribofuranose, and then you have the beta D-ribofuranose. Okay, now the anomer that is usually used within the ribose that is in in RNA, and of course, if we wanted to turn this into deoxyribose, all you'd have to do is take this alcohol group off and replace it with a hydrogen. Okay, so again, the optical isomerism of this first alcohol group in the deoxyribose within DNA is again that it's coming out of the page towards us. So basically, you always use the beta anomer in DNA and RNA. Okay, right. So, as I say, people will often just refer to beta D-ribofuranose as ribose. So when people say ribose, usually what they mean is beta D-ribofuranose. But strictly speaking, there are 4 different optical isomers of ribofuranose, and also you should really question whether it's the cyclic ribose that they're talking about or the linear ribose they're talking about. Okay, so if you turn this into the linear ribose and you cleave this bond here to do that, what happens is you only end up with 2 optical isomers. You end up with D-ribose and L-ribose. Okay, whereas once you've got the cyclic form, the ribofuranose, to end up with 4 optical isomers. You have D-ribofuranose and L-ribofuranose, but for each of those you then have the alpha and the beta anomer. So you have alpha D-ribofuranose, beta D-ribofuranose, alpha L-ribofuranose, and beta L-ribofuranose. Now, when people talk about ribose, as I've already said, they don't generally mean the linear forms, and they usually mean a cyclic form, and the form they usually mean is beta D-ribofuranose. Whew. Right. Okay, so now that we've discussed that, we are in a position to discuss what the enzyme amidophosphorybasyl transferase is going to do. So basically, it's going to act on an enzyme which is called alpha-phospho, okay, and then ribosyl pyrophosphate. So alpha-phosphorybasyl pyrophosphate. And for short, alpha-phosphorybasyl pyrophosphate is often abbreviated to alpha, and then it's P for phospho, R for ribosyl, and then PP for pyro. Oops, not like that. Okay, let me write that again. Okay, alpha, P, R, PP. Okay, I was trying to make it have a nice pattern there, but that is not correct. So alpha phosphoribosyl pyrophosphate. Now, this alpha is telling us which anomer of D-ribofuranose you are talking about. And I should stress that unless told otherwise, when someone is referring to ribose, you should assume that they are talking about D-ribofuranose. They should tell you maybe which optical isomer they're talking about. Okay, so here it is telling us which anomer they're talking about, i.e. whether they're talking about the alpha or the beta form, but it's assuming that you understand that they are not talking about L-ribofuranose, and also that they're not talking about the linear form. Okay, so what's the structure of this molecule? So I think we can squeeze it in here. So basically what you do is you have this ribose structure again, so I'll draw this here. Okay, and then let's put the alcohol groups on the second and third carbon, because we can always be sure of those. They are always fixed. If you change those, it's no longer ribose. Okay, now we know that this is the alpha form, so it's going away from us. Okay, but we won't draw it yet because we're going to have to take account of these other things in here. Okay, and then the fifth carbon, of course, we assume we're talking about D-ribofuranose. So the fifth carbon is coming out of the page towards us. Okay, and now off this fifth carbon's alcohol group here, what you're going to have is a phosphate group. So let me draw that here. Okay, so here is this phosphate group, like so, and so that's the phospho, by the way, the alpha phosphoribosyl. Phosphoribosyl is this phosphate group off this fifth carbon's alcohol group. Now, off the first carbon's alcohol group, which we know is going back into the page away from us because of the alpha here telling us that we're talking about the alpha anomer, you then have a pyrophosphate group coming off here, which remember is these two phosphate groups linked together. Okay, so here is the pyrophosphate group. So the whole thing now is this molecule alpha phosphoribosyl pyrophosphate, alpha PRPP. Right, so the amidophosphorybasyl transferase, which is the target for this drug azathioprine, is going to work on this molecule alpha phosphoribosyl pyrophosphate, and let me show you what it's going to convert it into. So it's going to convert it into a molecule that is called 5-beta riboxylamine. Okay, so what this is telling, oh whoops, sorry, no, I've missed a bit out. 5-beta phosphoribosyl riboxylamine. Okay, so let me write this again. So this is 5-beta and then phosphoriboxylamine. Okay, and you might like to try to attempt to draw this for yourself. Okay, right. So, firstly, let's start off with the ribose structure again, or rather the D-ribofuranose structure, strictly speaking. Okay, so here is this 5-membered ring. And then, of course, we assume we're talking about the D form, so we'll have the fifth carbon coming out of the page towards us. Okay, and off this fifth carbon, you'll then have an alcohol group, and we're told that off this fifth carbon we have a phosphate group here. So we'll leave that phosphate group that we had on alpha phosphoribosyl pyrophosphate. We'll leave that there, basically. Okay, so that hasn't changed at all from alpha phosphoribosyl pyrophosphate. Okay, and then off this third carbon, everything still remains the same. This alcohol group goes back into the page away from us, and off the second carbon this alcohol group goes back into the page away from us. And now, we're talking about the beta anomer now. Okay, so it's changed. This group now comes out of the page towards us, and it's even changed completely in its structure. It's no longer an alcohol group because we're talking about riboxylamine. Okay, and that means that we now have an amine group off here. So this is what this drug, what this enzyme, amidophosphorybosyl transferase, is going to do. It's going to convert alpha-phosphoribosyl pyrophosphate into 5-beta phosphoriboxylamine. Okay, now how is it going to do this? Well, basically, it's going to take in water and glutamine in order to perform this conversion. Right, so this is going to be quite difficult to show because the pictures are on the opposite sides. Okay, but we'll do our best. In fact, actually, I might just redraw out the alpha phosphoribosyl pyrophosphate molecule because I think it is going to be too difficult to show it over the page. Alright, okay, so remember, this bit has not changed in the slightest up here, so the 5th carbon still comes out of the page towards us. Okay, it then has an alcohol group off it which has been linked to a phosphate group via a phosphoester link. Okay, then of the 2nd and 3rd carbons, the alcohol groups are forced to go backwards. Okay, otherwise it would not be ribose. It would be some other aldopentose. Okay, and then the alcohol group off here did go back into the page, and then it had this pyrophosphate linked off it, like so. Okay, right. So, how are we going to convert this into this here? Right, this is what the enzyme amidophosphorybosyl transferase is going to do. Okay, so, basically, one of the things that we're going to bring in is water. Okay, and what you can imagine water doing is hydrolyzing this bond here. This is not supposed to be a molecular flow diagram, basically. I'm not going to show you actually how the reaction occurs. I'm going to show you how to make sense of the overall reaction. Okay, so what you can imagine happening in order to keep track of the reaction for yourself is this water coming in and hydrolyzing this phosphoester bond so that you end up with an alcohol group there and also a pyrophosphate molecule that's been given off. Okay, so let me put this here. Okay, so one of the products you're going to get will be a pyrophosphate molecule, like so. And I'll stress that I'm not showing you the molecular mechanism. I'm showing you how the reaction makes sense. So on this side of the equation you've got water, on this side of the equation you've also got pyrophosphate. So you can imagine the water coming in here, hydrolyzing this phosphoester link here, producing this pyrophosphate molecule, and also producing an alcohol group back here. Now, we need to exchange that alcohol group for an amine group, and also we need to alter the optical isomerism. So where are we going to get that amine group from? Well, basically we're going to get it from the amino acid glutamine. So let me show you the structure of the amino acid glutamine. Okay, so here is the amino group of the amino acid, here is the alpha carbon, here is the carboxylic acid group. So I've started by showing the core structure of the amino acid, and now the R group of glutamine, you have 2 methylene groups, so I'll put these here, 1, 2, and then you have a primary amide group, so you have this group like so, a carboxylic acid group which has been linked to an amino group like so. So this is the amino acid glutamine. Okay, and the single letter amino acid code for glutamine is Q. Right, okay, so basically what we're going to do is chop off this amino group from the glutamine. We're going to chop off the alcohol group from the intermediate that we've imagined forming. Remember, we've imagined that we've chopped off the pyrophosphate with this water, and we've imagined now that we've got this intermediate where we just have the alpha phosphoribosyl, basically, or the alpha phosphoribose, I suppose you'd call it. Right, and we're then imagining we're taking the alcohol group off, we're taking the amino group off, and we're swapping them. We're putting the amino group on here, and we're putting the alcohol group on here, and then obviously we'll alter around with the optical isomerism here so that the amino group comes out of the page, and the hydrogen that also comes off this first carbon goes into the page. Okay, so the other product of this reaction will be the glutamine, which has been replaced with an alcohol group there. So that's the amino acid glutamate. So let me just draw you the structure of the amino acid glutamate. So here is the amino group here. Here is the alpha carbon again. Here's the carboxylic acid group, like so. And then the R group of glutamate is that you have these two methylene groups again. 1, 2. And then you have a carboxylic acid group now here, because the amino group's been taken off and replaced by an alcohol group. So this is glutamate, or strictly speaking, what I've drawn is glutamic acid. Glutamate would be the conjugate base of glutamic acid. It would be the molecule once it's given away its proton. Okay, so of course in a solution containing glutamic acid, it will be in equilibrium with its conjugate base, the glutamate. So that's why people use the two words interchangeably. But what I've strictly drawn there is glutamic acid, so I should put that. And the single letter amino acid code for glutamic acid is E. Right, so basically this enzyme, which is amidophosphoribosyl transferase, the ATAs, is going to convert alpha phosphoribosyl pyrophosphate with water, with glutamine, into this 5-beta phosphoribosylamine plus glutamic acid plus pyrophosphate here. Okay, so that's what this enzyme catalyzes. Why is this so important? Well, basically this is a precursor for production of purine nucleosides, basically. Okay, well, nucleoside monophosphates. So purine nucleoside monophosphates. Okay, so what does a purine mean? Well, basically some of the organic bases are considered purines, and I'll show you those just in a moment. So purine nucleoside monophosphates. So basically adenine and guanine organic bases are considered purines because they have purine rings within their structures. Okay, whilst where did I put this? Here it is. Whilst the others, cytosine, thymine, and uracil, those are considered pyrimidines because they only have pyrimidine rings within their structures. So, in order to produce the guanine and adenine nucleoside monophosphate, which remember means ribose bound to those organic bases, bound to phosphate groups, you require the synthesis of this molecule 5-beta phosphoribosylamine. Okay, so you're now going to stick the purine organic bases onto here to create the purine nucleoside monophosphates. Okay, and those are then needed to create the purine nucleoside triphosphates. Okay, so all you do is stick 2 more phosphate groups on it then to get the nucleoside triphosphate, and you also need this to create the deoxynucleoside triphosphate, for which you're obviously going to have to stick 2 phosphate groups on and also remove this alcohol group off the second one. And of course you need those purine nucleoside triphosphates and purine deoxynucleoside triphosphates in order to synthesize mRNA and DNA respectively. Okay, so if you block this, you block the synthesis of the purine nucleoside triphosphates, which blocks mRNA production, because of course you need them. You know, they make up half of the organic bases in mRNA. If you don't have them, you're going to get stuck. Okay, and blocking the production of deoxynucleoside triphosphates is going to stop the production of DNA, okay, which therefore is going to stop DNA replication. So both of those things are going to contribute to preventing cell division, basically. Okay, so that is how azathioprine blocks cell cell division, but this doesn't explain why that won't occur in every single cell within your body. Okay, and this will occur in every single cell in your body. It will block the amidophosphoribosyl transferase in every cell in your body. The difference between T and B lymphocytes and other cells in your body is that other cells in your body have alternative mechanisms for creating the purine nucleoside triphosphates and the purine deoxynucleoside triphosphates, whereas T and B lymphocytes, this is their only pathway basically, and once you cut that off, you stop all synthesis of those purine nucleoside triphosphates and purine deoxynucleoside triphosphates. Okay, and that's why it has such a marked effect on T and B lymphocytes and stops cell division within those B and T lymphocytes and therefore produces immunosuppression. Okay, in the next video we'll then turn our attention to mycophenolate.